Salam sejahtera to Dr. Asma. In this video, I would like to present about the recombinant bovine growth hormone in stimulating fish growth. These are the contents that will be included in this presentation. First, introduction. So let's take a look on growth hormone. The growth hormone of vertebrates are produced primarily by pituitary under the manipulation of hypothalamic factors and also environmental factors. Stimulatory and inhibitory hypothalamic factors such as ghrelin and somatostatin are playing roles in regulating the production and release of growth hormone from pituitary. Environmental factors such as temperature, food availability, and photoperiods also affect the secretion of growth hormone. Growth hormone is acting as a growth stimulator directly to the on specific receptors possessed by some tissues such as muscle, intestine, and kidney, and indirectly by way of inducing the generation and synthesis of insulin growth-like factors to promote growth in vertebrates, including fish. Recombinant growth hormone is a synthetically produced hormone that mimics the naturally occurring growth hormone in fish, which playing a role in stimulating the growth of fish. To produce recombinant bovine growth hormone, the process typically involves isolating the gene coding for bovine growth hormone from the pituitary gland of the cattle. This gene is then inserted or cloned into a suitable expression vector, such as plasmid vector. Restriction enzymes are used to cut both plasmid and bovine growth hormone gene to create a compatible ends which enable the gene to be inserted into plasmid. Ligase is applied to seal the gap in DNA backbone, which forming recombinant plasmid. The vector serves as carrier to clone the gene into host cells, which are normally bacteria, E. coli, or yeast, through transformation. Methods such as electroporation is used to promote the uptake of the plasmid by host cell. The host cell was plated on agar plate that consists that consisted of antibiotic, and only the bacteria successfully taken out the plasmid that carried the gene of antibiotic resistance will grow well. The recombinant DNA is transcribed into mRNA within the host cell and translated into protein. The host cell will utilize the cellular machinery to express recombinant growth hormone. After production, the host cell that consisted of expressed recombinant growth hormone will harvested. To harvest the host cell, methods such as centrifugation or filtration are applied. Next, the harvested cells are lysed to release recombinant hormone. After that, the hormone is purified using biochemical techniques such as chromatography to ensure it meets purity and safety standards. This recombinant bovine growth hormone can then be administered to farm fish either through several methods to enhance growth rates and improve overall productivity in aquaculture operation. This is a simple figure that shows the process of producing recombinant growth hormone. These are the six importance of recombinant bovine growth hormone in aquaculture sector. So the first one, increase fish appetite. Secondly, increase hyperosmoregulatory ability, followed by enhanced food conversion efficiency of fish, stimulate tissue growth, optimize production efficiency, and also reduce the culture period. Then, I would like to continue with delivery methods of recombinant bovine growth hormone to fish. So the first method is injection. It is time consuming and require high labor power. And the second method is immersion. It is only effective during the larvae or juvenile stage of fish, and it will cause stress to fish as treatment usually requires several times to complete. And the lastly is oral delivery, which through the enrichment of feed. It is widely practiced in aquaculture as it is time efficient. and fish was treated by the recombinant growth hormone when they consumed the enriched pellets. Now, let's move on the first study case. So the culture species is Lithovenus vaname, which commonly known as violet shrimp. The oral administration of recombinant bovine growth hormone to vaname shrimp significantly increased the weight gain rate, body length, and survival rate of the juvenile vaname shrimp. The base ratio of recombinant bovine growth hormone being applied to increase the growth performance of vaname shrimp is 0.07% by weight of the recombinant bovine growth hormone based on 100% by weight of the total shrimp feed composition. This study case showed that the shrimp fed with recombinant bovine growth hormone treated pellet presents 349.2% of weight gain rate, 46.4% of body length, and 21% of survival rate higher than control group, which fed with untreated pellet only. Second study case, the cultured species was Oncorhynchus mycis, which is rainbow trout. Oral administration of 1% recombinant bovine growth hormone to rainbow trout significantly induced the appetite, mean total length, and mean average daily length, and also average daily growth of the rainbow trout after 84 days culture period. The rainbow trout treated with recombinant bovine growth hormone consumed 6.95% more feed aggressively than control group. The mean total length of treated group was 3.46% higher than control group. Besides, the mean average daily length of treated group was 18.42% more than control group. The average daily growth of treated group was 9.1% higher than the control group. For the last study case, the cultured species was Italurus punctatus, which is channel catfish. Injection administration of 10 microgram recombinant bovine growth hormone to channel catfish fingerlings significantly induced the growth performance. The mean growth rate of channel catfish fingerlings treated with recombinant bovine growth hormone were 10% higher than the control group. Then, the condition factor of treated group was higher than the untreated group. And for the stimulation in appetite was observed in treated group. The amount of dry feed consumed as an average 929% of body weight per day was obtained in treated group as compared with 728% from the untreated one. There are 4 points in challenges and limitations of recombinant bovine growth hormone. The first one, market acceptance. Consumers will wary of buying the fish that treated with recombinant bovine growth hormone. It will affect the market demand, thus reduce the profitability of aquaculture sectors. And the second one, environmental impact. The hidden capability for recombinant bovine growth hormone to enter the natural aquatic ecosystem, which poses the risk of affect non-targeted species. And the third one, antibiotic residue. Antibiotic residue poses the risk in affecting human health is a major concern. As example, antibiotics are often involved in preventing bacterial contamination during processing of recombinant bovine growth hormone. It might cause development of antibiotic-resistant bacteria. And the last one, production cost. Producing recombinant bovine growth hormone required specialized facilities due to the complicated biotechnological process. It is not friendly, especially to small-scale aquaculture operations, in either producing the recombinant bovine growth hormone by themselves or purchasing from other suppliers. After that, recommendation. So the first, more research is needed to fully understand the long-term effects of recombinant bovine growth hormone used on animal welfare and human health in order to address regulatory restrictions. And second, exploring other new alternative delivery methods such as slow-release formulations to improve the practicality and cost-effectiveness of recombinant bovine growth hormone use in aquaculture. Lastly, I would like to end with conclusion. In conclusion, recombinant bovine growth hormone is an effective biotechnological method in promoting the growth of aquatic organisms and also optimizing the profit in aquaculture sector. These are the references that I use to complete this presentation. That's all from me. Thank you and have a nice day.